Hello everybody, so I'm going to take the next 10 minutes or so just to tell you a little bit about what's coming along for CML and what might be suitable for some people that are perhaps not doing so well that they might be able to discuss with their consultants. So within the UK we have the National Cancer Research Institute, and these are the clinical trials that are organised through that organisation. We have a number of studies in setup, a couple of studies recruiting, and then there are some other studies in follow-up. So in terms of the studies in setup, I'm going to tell you quite a lot today about Taster, which will hopefully be open before the end of the year. I'll tell you a bit about the CALLS study, which is open just now. There's the ABLE 001 study, which you may have heard of. And then there's this new drug, KO706. And in terms of the studies and follow-up, I'm not going to spend so much time on these. I will talk a little bit about Matchpoint, which was our blast phase study, and Optic and Before are now closed. So Matchpoint was a study for patients who had blast phase CML. So for those patients that either presented or progressed to the most aggressive phase of CML. And it was a trial in which we combined intensive chemotherapy with ponatinib. So it was a phase 1 study, so it was a very early phase study because this therapeutic approach hadn't been tested before in CML. And we recruited 17 patients. The study closed to recruitment last May. The results are looking quite promising. The patients had 2 cycles of chemotherapy with ponatinib, then went on to have a transplant, and then went back onto ponatinib after the transplant. So the results do look quite promising, and they will be presented later in the year. So hopefully we'll be able to put them on the CML support website later in the year. And the design of the trial looks at both how effective the drugs are and also how safe the drugs are. And we're using that to identify what the safest dose and most effective dose of ponatinib is to take forward into future studies in blast phase CML when you're combining it with intensive chemotherapy. So I think I've probably just said all this. The important thing is that when patients went into the study, they all had 30 milligrams of ponatinib, so it's not the full dose of ponatinib, it's a little bit less. Once they'd had their transplant and were in remission, they then went on to 15 milligrams, which is the lowest dose of ponatinib, and patients maintained their responses on that dose of ponatinib, which is important to know. We didn't see any significant safety concerns that we wouldn't expect with the chemotherapy alone, so the adding in the ponatinib didn't seem to have any additional toxic effect, and specifically we didn't see additional problems with infections or cardiac toxicity. So the next study I'm going to talk about, and this has gone a bit strange, is the CALLS study, which is for doing mutation screening for patients that aren't doing as well. So patients that don't have a major molecular omission may be eligible to go into this study, and it's open at a number of sites around the UK. The study's recruiting really well, and it's for patients with any phase of CML that their consultant's concerned they may have a mutation, and also for patients with another acute form of leukemia called Philadelphia chromosome-positive acute lymphoblastic leukemia. It can be for patients who are on their first-line treatment or any subsequent line of treatment, and it's comparing different ways of measuring the mutations. So it's comparing something called Sanger sequencing, which is the standard method for detecting mutations, which has a sensitivity down to about 20%, compared to next generation sequencing, which has much more sensitive and will detect much smaller levels of mutations, which may be causing resistance in patients. So this will start to report over the next couple of years. It is going to be recruiting probably for at least another year. So this may be a study that some people are already involved with, and if not, you may get asked to participate. It's not changing your treatment specifically, and it's not offering a new treatment, it's just offering a new test that may guide the treatment that your consultant gives further down the line. So this is the eligibility for the CALL study. So patients must have either a warning or failure, so not in an optimal response, as I think Jenny and Jane discussed earlier, and they must not be in major molecular remission. And the reason for that is that if you've only got a tiny level of the BCR-ABL there, the test isn't sensitive enough to detect very, very low levels of the mutation. Patients that have had previous kinase domain mutation tests can still go into the study. It doesn't matter what the test result was. And patients who have newly switched TKIs due to a suboptimal response can go into the study. So it doesn't matter if you're already changed drug, you can still go into the study. So the next trial I wanted to talk about was TASTER, which is our new study for resistance patients, which I would expect to be open later this year. It's funded by the Stand Up to Cancer campaign via CRUK. So this is a trial design that was developed by Tessa Hollyoak, who sadly passed away a couple of years ago. But this is really her legacy to CML patients within the UK. And this is for patients with chronic or accelerated phase CML who have failure. So either BCR-ABL above 10% after 6 months on a TKI or 1% after 12 months on a second or third generation drug. And it's for patients that are not at that time suitable for a transplant, and whether or not they have a response, they may be able to go into transplant in the future. That's acceptable. At the moment, we've got 3 arms for the study. We're aiming to open over 16 centres and recruit over 6 years. And when the study starts, we're going to have 3 arms. We're going to have a TKI-only arm, so people going in would stay on their current TKI, or they would go on to a TKI plus a new drug called an EZH2 inhibitor, or another new drug called an HDM2 inhibitor. And the EZH2 inhibitor and the HDM2 inhibitor have both been used in other types of leukemia, but they haven't been used before in combination with TKIs. So what we're looking for is a well-tolerated drug that will induce an improvement in BCR-ABL levels that we can then take forward into more patients to try and improve responses. For those patients that draw the control arm, these patients will have the option to switch to one of the different arms of the study after 6 months. So for patients going into the study, if they don't respond either to the control within the study or the first of the experimental arms, they can try an alternative arm within the study. So the TASTER clinical trial is not just a clinical trial, it has all these other work packages where we're trying to understand how the leukemia stem cells change with the treatment, so that we're hoping in the second half of the study we might be able to understand better the responses to the different treatments within the stem cells within the bone marrow, and then use that to predict which would be the best agent to add into the TKI. So the trial design looks very complicated, and you probably can't really see this from the back of the room, but for every patient that goes into the control arm, 1 patient will go into each of the experimental arms. And we assess the response after about 8 months. And as I said, for those patients that aren't responding on either the control arm or one of the experimental arms, they have the option to either come out of the study or to switch to the other experimental arm. And for patients obtaining benefit, they would continue on the drug combination for as long as they maintained benefit, but for a minimum of 81 weeks, so getting on for 2 years. The other studies I want to talk about, there's the ASSEMBLE study, the ABLE-001 study. This is a phase 3 study recruiting 222 patients worldwide. It's open at sites in the UK, the Hammersmith, Cardiff, Oxford, Liverpool and Glasgow. And it's randomising 2 patients to asciminib, which is the new drug, versus bosutinib, which I'm sure many of you have heard of. And the main goal of the study is to compare major molecular response rate at 24 and 96 weeks of the 2 different drugs. The main inclusion and exclusion criteria are it's for patients with chronic phase CML who have failed at least 2 of the ATP binding site TKIs and have a BCR-ABL level above 1% at screening, or for patients that are intolerant, it needs to be above 0.1%. And they shouldn't have had any prior therapy with asciminib or bosutinib. And the study's been recruiting pretty well. It's recruited really quite well in the UK and will close for recruitment probably towards the end of October this year. In terms of other studies, there's the K0706 study, which is a phase 1 study of a conventional tyrosine kinase inhibitor, so a drug like bosutinib or ponatinib, but it has T315 activity, and this is a study that's open at King's and at the Hammersmith. The other study is the ABL001 E2201 study, which is open at Hammersmith, Liverpool and Oxford. And this is a combination of imatinib with asciminib in patients with levels of BCR-ABL of between 0.01 and 1% after two years of first-line imatinib. And it's looking at adding a different dose of asciminib onto the imatinib or doing a straight switch to nilotinib. And it's looking to see which of these approaches is the best at obtaining a deep molecular remission. So that's everything I wanted to talk about. I'm happy to take any questions or catch up with people during the panel at the end.